Hello, everyone. We are the JU-Krakow team from Poland, and today it is our pleasure to present to you our project called PrymDetect.
Imagine standing by a river, once full of life, now devastated. In the summer of 2022, the Oder River faced one of the worst environmental crises in European river history. The victims? Hundreds of tons of fish that washed up dead on its shore. The communities surrounding the river were left in confusion and fear as misinformation about the causes of the disaster spread in media. Was it pollution? A chemical spill? As the mystery unfolded, the truth finally surfaced. The cause was Prymnesium parvum, also known as golden algae. It is a harmful algal species capable of forming toxic blooms, leading to large-scale fish kills. Throughout our research and deep engagement with the community, we uncovered the heart of the problem, detecting this algal species in water samples quickly, reliably, and right at the source, where time and accuracy mean everything.
During our iGEM season, our team took on the role of detectives as we decided to tackle this challenge. To achieve our goal to detect Prymnesium parvum DNA in water samples, we decided to apply the Sherlock method. Here's how it works. Sherlock is a diagnostic tool that utilizes proteins from the Cas family, like Cas13a, to detect nucleic acids, in our case, the DNA of Prymnesium parvum. A specific guide RNA, called crRNA, is designed to target the sequence of interest. So what does it look like to apply this method to detect Prymnesium parvum? After collecting the water sample, we isolate the genomic DNA. Next, we amplify the DNA using recombinase polymerase amplification, which proceeds at 37 degrees. After amplification, we add all the Sherlock reaction components, which include T7 RNA polymerase, which transcribes the target's DNA into RNA. Once Cas13 binds to the target RNA, it exhibits a collateral RNase activity, non-specifically cleaving single-stranded RNAs around it. This activity is harnessed in the assay by using synthetic RNA reporters. For standard fluorescence detection, we use an RNA reporter labeled with a fluorophore and quencher. If we see a fluorescence signal, that indicates that some reporters have been cleaved, meaning that the DNA target is present in the sample. This method requires a plate reader, so it's best suited for laboratory applications. For field use, a simpler method is the lateral flow test, which involves the same methodology, but the result is read using a simple strip, such as those you may know from COVID-19 at-home tests.
As part of our contribution for future iGEM teams willing to work with Sherlock and purify the Cas13a protein, we simplified the currently available protocol. After expressing our protein in the E. coli, we developed a three-step purification protocol. The first step was IMAC, immobilized methyl affinity chromatography, where we captured the protein on a NINTA resin. Then, half of the fractions were digested using the sumo protease to assess the impact of the TAC removal on the protein's function, stability, and structure. We decided to do it as nobody has done it before, and sumo protease is quite costly to be used during this protocol. Then, using an ECTA system, we performed ion exchange chromatography, so second purification step. Finally, we obtained pure Cas13a protein after the size exclusion chromatography step. Then we performed activity tests to compare two versions of the protein, digested and undigested. We haven't seen any significant differences. We also examined structural differences using the cyclodichroism, also no differences were observed, and thermal stability within the nano-DSF. So we've shown that TAC removal isn't necessary, and this means that the future purification efforts, we can skip this step and make the purification protocol more cost-effective and time-efficient.
Additionally, what's unique about our project is that we worked with an understudied algal species and established protocol for culturing this organism. Our insights can be valuable for future iGEM teams, such as you, who would want to work with Prymnesium parvum to help mitigate its negative environmental risk. Since Prymnesium parvum is a non-model organism, we had to start completely from scratch and cultivate it from water samples obtained near the Oder River, where the blooms had been reported. We tried different techniques to recreate the natural habitat of the algae as closely as we could, keeping track of details such as salinity, pH, and lightning.
Our project couldn't be successful without IT support and bioinformatics. It's a little bit like Sherlock Holmes trying to tackle a mountain of data with just a magnifying glass. Even he would struggle without the right tools. So I mentioned data, so let's talk data. Unfortunately, there were no available sequences for the strain of golden algae from the Oder River. So we sent a fragment of isolated DNA for sequencing. Blast analysis revealed that our fragment was similar to several Prymnesium parvum strains in the database. We chose the KAC39 sequence as a proximity that we liked to continue with. KAC39 is a strain which produces type B toxins and originates from Norway. We identified key features in the KAC39 sequence, such as the internal transcribed spacers, or ITS for short. So, what are the ITS sequences? ITS sequences are non-coding regions located between the coding sequences of ribosomal rRNA genes. ITS sequences are really cool and really useful. Because of their documented variability between species, we saw them as perfect target for our test, since targeting these regions would allow us to specifically detect Prymnesium parvum and none other, even closely related algae.
Finally, all the previously developed elements came together to create a precise and accurate tool for detecting Prymnesium parvum. We designed and tested 18 combinations of cRNAs and primers, and we selected and further characterized the ones that performed most efficiently. Next, we moved on to developing the lateral flow assay for usage in the field. So how exactly does the lateral flow assay procedure work? Here, we use RNA reporters labeled with FAM on one end and biotin on the other. If Prymnesium is absent in the sample, the C line on the test captures the intact biotin-labeled reporters, leading to only the lower C line strip being visible. However, when the Prymnesium is present in the sample, Cas protein cleaves the reporters, and gold nanoparticles with anti-FAM antibodies migrate further up the strip, producing a visible T-line, which indicates a positive result. We successfully adapted our optimized Sherlock components for use with the lateral flow assay and demonstrated that this method reliably detects Prymnesium parvum DNA. We also aimed to simplify the procedure as much as possible. We found that both reaction steps, RPA and the Sherlock readout, can be incubated at 37 degrees using body heat, such as by the user holding the test tube in their hand. Overall, our results confirm that we have successfully created a test that can be used to quickly and efficiently screen water bodies for the presence of Prymnesium parvum.
The challenges of the 21st century, such as the pandemic or the increasing number of harmful algal blooms, highlight the urgent need to develop tests to quickly identify dangerous factors in our environment. We believe that standardization, which lies at the heart of synthetic biology, can help achieve this goal. That is why we created SynLog, the cRNA synthesis system for Sherlock. Our goal here was to make detection using Cas13 proteins more accessible to future iGEM teams and scientists worldwide, regardless of the factor that they want to detect. The cRNA is the molecule responsible for the assay's specificity. It includes a DR loop and a spacer complementary to our detection target. When generating cRNA from DNA templates, a T7 promoter is appended to the cRNA sequence, and the whole construct is ordered as a DNA reverse complement. Our system introduces a fragment called a cassette, which already consists of the T7 promoter and the DR sequence. It enables scientists to only design the short sequence that they want to target. The cassette also includes a chromoprotein reporter assembled from basic parts, which allows for easy insertion of custom spacers and straightforward screening of the correct colonies. Using our platform, we successfully obtained all of the CRRNAs we designed for detection of Prymnesium parvum, as well as a control construct. We believe that our highly standardized and modularized system has strong potential to be a building block in creating reliable detection tools by future iGEM teams and scientists around the world.
We also thought about alternative readout methods for the Sherlock assay. So we created the PrymChip, a 3D printed device designed to detect fluorescent signals and translate them into concentrations of the RNA probe. We saw this device as a reusable solution for repetitive long-term monitoring of different water bodies. After numerous iterations and improvements, we finalized the PrymChip. PrymChip is now composed of 3 main parts: a black detection box, a yellow electronic box, and a purple toolbox. So, how does the prototype work? Some lucky people that visited our booth may already know something about it, but let's walk through it. The detection begins with the toolbox. First, we use a syringe to collect the sample and transfer it into the detection chamber. After closing the box with a black lid, we turn on the blue LEDs to excite the green fluorescence signal. Next, we place a smartphone on the PrymChip and take a photo of the chamber. Our Python software measures the green fluorescence signal to estimate the concentration of fluorescein in the sample. The fluorescein serves as an example of a molecule that mimics the actual Sherlock RNA probe. The PrymChip is an incredibly universal device where you can swap out filters and diodes, use your own pictures with our code, or even completely redesign the toolbox. Now, as a well-equipped team of detectives, we are able to assist all kinds of users to carry out their own investigations to locate the culprit.
As part of our integrated human practices, we connected with so many people. First and foremost, the fishermen, who help us identify the most urgent aspects of the Oder River disaster. Secondly, expert on the parliamentary team for the Oder River, who confirmed our concerns about inadequate water body monitoring and supported our idea of the PrymDetect to be valuable for quickly screening multiple water bodies. And we also consulted academic experts on basically every step of our project and integrated their feedback to refine it.
But the danger hasn't passed, and despite the efforts of dedicated institutions, the blooms keep reoccurring since the initial disaster. Money was spent and time passed. Still, they weren't able to find a solution. But we were. Our team has been invited to consult with the Polish Ministry of Environment, who are potentially interested in implementing our solution. Additionally, our project will be continued at the Laboratory of Metabolomics at the Jagiellonian University. As the first ever iGEM team from our university, we did not expect to make it to this stage today. Our project was completely planned and carried out by our 10 student team members, all first-time iGEMers. We are proud and happy to see our project making a meaningful impact in Poland, around the world, and especially in iGEM. Thank you.
Woo! Thank you guys so much. And congrats on being first-time iGEMers. That's amazing.